Alright, so what we're doing now is cell communication part 2, and these are signal transduction pathways. We've already mentioned the fact that cells have to communicate, and I'm sitting here looking at a typo. Don't worry about it. Anyway, and we've talked about the fact that sometimes it is strict cell-to-cell communication. Sometimes it is a short distance, such as the neurotransmitters between the synapses. And then the other one, of course, was the long distance, sending out hormone signals. Now, the next part about this is that we need to figure out what we're going to do with these signals. In this next slide, which hopefully is coming up, what we're looking at is an analogy. This is an old-fashioned telephone, of course, and this is where you would speak into, and then this part was the ear part. I think you can recognize that. But you could unscrew these things. And what the deal was is that I always kind of wondered when I was your age, how in the heck do they do this? Because you're speaking into that bottom part. So what the deal is that you're going to turn electrical sound waves into electrical energy and then back again. And so what we're talking about here, this is called transduction. And so that's the name for the part that was right here in this part of the phone and this part of the phone. They were called transducers. All right, anything. Your cell phone has the same thing, but it's, of course, a little tiny chip in comparison. Now, same thing happens in a cell. These are some of the big-time names, gang. Unfortunately, this is one of the enduring standards of AP Biology. I think it's enduring standard number 3. You will need to know these processes. If you can just remember a couple of them, we're okay. Just when I say sketchy part, I mean that you've got to know the general idea. All right. So the process by which a signal on a cell surface is converted into a specific cellular response is a series of steps called a signal transduction pathway. Now, you've already done biochemical pathways. That's photosynthesis and cellular respiration. So you know that usually what that means is one thing makes the other occur, and hopefully things don't go backwards. You don't want that to happen. This is the actual picture from your book and stuff like that. There are 3 things that have to occur when the signal's going out. Let's talk first. Let's say that, for example, somebody comes up and scares you, and you have to run really, really fast. Well, what happens is that the adrenal glands are going to send out some chemicals, and they're going to tell your bodies to start breaking down glycogen into glucose so you can make ATP so you can run. All right, so the first thing you have to have, of course, is reception. This is where your cell's going to actually have something on the surface that's waiting for this chemical to come in, and it will bind to it. Then what you have to do is have the transduction, or changing it from one signal into the next. For example, here's what we're talking about. This is going to be phosphorylase. Anyway, sometimes it's just the one simple thing. It opens up a channel and lets it through. Other times it's got to go through what we call a conformational change, and you'll see that in a minute. And then last but by no means least, once you get the signal, what do you do with it? You want a response. And so that's the part we're going to look at right now. All right, the next slide that I've got is going to go, I'm going to have to go to the internet here, and Wiley has a really good animation on this. And we're going to pretend that I can get this easily. And we're going to say copy. Or I can say open hyperlink. Where's my little computer, people? All right. And so here comes our internet little figure here. Let me resize my screen. And here we go. All right. What you're looking at here, what they're trying to tell you is, here's the brain, and you see a bear. So what do you need to do? It is time to go. Let's run. And that's why I picked epinephrine as the thing to break down glycogen. All right. So what we're looking at on this next part is, let's say this is your target cell, okay? Here's the receptor waiting for something to come in, okay? So you send out, the adrenal glands send out the epinephrine, and there we go. All right, here it comes. Boom. And you will notice that what we've had happen here is something. This turned around, grabbed these, and then something occurred right there. So what happens here, we're going to say a protein will enter the nucleus, is going to bind to the DNA strain. I know you all know what that looks, and it's going to affect transcription. Well, since you haven't had that part yet, transcription is the making of proteins. And a new protein can be made from the messenger RNA that will alter the cell function. All right, so you watch it, it goes, and that was a piece of messenger RNA, and because it's single-stranded. And then here you'll notice it's going to altered cellular function. On the next one, another scenario, the cell needs to keep it in there. And so our protein will now remain in the cell, and it affects cellular metabolism. So it remains right in here, and it's only going to affect what's happening inside the cell. Okay. Now, another example here is we have the hormone. Here's an endocrine gland way up here. And then we're going to come down here to the bloodstream. It's going to go to the target cell. All right. And this is how you're going to get these actual, what we're going to call the signal. This is part of that signal transduction pathway. All right. What the deal is that most hormones can't go across that membrane. Don't forget, you've got integral proteins, you've got peripheral proteins, you have ion channels, but most hormones are fairly large and they're not going to be able to get across, or their chemical makeup doesn't allow them to. So what happens is that you noticed it bounced. Okay, if I go back, you can watch it again. Bounce, oh, won't let me in. So what do I do? What I'm going to do is go to a transmembrane protein. Now you'll notice right there, a little bit of a conformational change. Just the word conformations means shape. Okay? Then what happens is that signal can be actually picked up and then it's turned on inside the cell membrane. Don't worry about the GDP and stuff right now in GTP, although you will notice that a phosphate was picked up. All right? So what will happen is the hormone signal can lead to what's called a cascade of modifications that will either activate or you may need to inactivate something. All right, please watch what happens. And we're going to watch this hormone come down here. We're going to do this. We're going to change the shape. It's going to pick it up. And you see where that says turned off? Watch what's going to happen. And this is in your bloodstream. Boom, shape change, move over, turn on. Since that turned on, I'll turn that on, and then I'll turn that on. That's the term cascade. All right, and of course, this is evolutionarily speaking, in particularly multicellular organisms, very important. All right, so where we are right now, we're going to talk about epinephrine. It's adrenaline. This increases the readiness of the muscle cells by mobilizing energy stored in the form of glycogen. And when you have an environmental stress, someone comes up and shocks you or scares you, or you see a bear, epinephrine is released from the adrenal glands. Now watch what happens. What we're going to have happen now is that there's the change. This is the G protein going and okay, turned on. All right, and it binds GTP and releases GDP. That's the phosphate group. And it moves over there. And this little thing is called adenylate cyclase. Do you have to remember that? Nope, that's what I was telling you. Anyway, watch what happens here. This little thing has to be turned on. All right, and it's turned on, if you'll notice, by taking and using ATP. But rather than leaving ADP, what you're going to see is that we have left CAMP. This is what we call CAMP, or cyclic AMP. And this is our secondary messenger. 2 phosphate groups are gone. When you did cellular respiration, you kind of wonder where all those phosphates come from. You have lots and lots in the cell because of these things. Now watch. There's a picture of the circular shape of it. All right. What CAMP does is it's going to bind and activate a protein kinase. Now you have to have the CAMP, and this one you need to remember. Cyclic AMP. It turns it on. You can already see what's going to happen. This activated kinase, and remember, ACE means proteins. These are what transfer phosphates from ATP to other proteins, and you've already heard this called phosphorylation. Some will activate, some will inactivate. Excuse me. Anyway, protein kinase is going to go over here to our intracellular protein that is going to actually produce the breakdown of glycogen into little glucose units. Notice, for one piece of hormone, one piece of epinephrine, one piece of adrenaline, we were able to break down this chain. This is called amplification. All right. Some receptors turn them off. You'll notice, watch this. All right, what we've done at this point over here, you'll notice it's on. We changed the shape right there. In goes our G protein, which is going to then release. For that, we're going to take this activated here, but since it's already on, it's going to deactivate it, and it's off. All right. Some secondary messengers will move into the nucleus and enhance gene expression. In other words, they're making, watch all the different pieces of messenger RNA that keep going. All right, that's another form of amplification. One hormone can lead to production of many of these CAMP molecules. Boom, 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 boom. Now, when we talked about diffusion and concentration gradients and stuff like that, this is part of what that was all about. In other words, when you start to get a buildup and you'll change the way your concentration gradient is, that's what will turn things off and on. All right. Steroids can enter the cells without the help of receptors. That's because they are a fat, and of course you've got your lipid membrane, lipid membrane there. And this goes through a bunch of quizzes and stuff. You can do that later if you want to look at these. All right, on diseases, this is an example of one that you might have heard of in WAP or maybe even APUSH. This is called cholera, and you have massive diarrhea. And whenever you have massive diarrhea, you know that you're losing a lot of water, and it could possibly be, it's potentially dangerous because you could dehydrate and then you'll die. All right, what happens here is that the G proteins, which normally hydrolyze, and that means water splits, their GTP back to GDP. All right, that's the normal. Okay, notice that we've taken those off. Cholera toxin modifies it so that that phosphate group cannot be hydrolyzed. And so that water, which would be there to do that, is no longer being used, and it starts to build up, and then you have your problem. So another example would be pertussis. This is whooping cough. It will prevent the G protein from binding, and so what you've got is an overactive type of thing. Watch this one. See, it won't go through and let it do anything, and so we're just making and making and making a modified. I'm sorry. We're just making and making the it inhibits the receptor pathways in the respiratory tract. Told you. Still trying. All right. And we'll have to quit here in a minute. This one is the thing that the same type of thing will turn on cancer genes. You talked about uncontrolled growth, and if you haven't, you will. And so these small proteins, it's called the RAS gene, because what they'll do is they can activate cell growth processes, and that's called a tumor. At levels, at certain areas, you can have drug interventions and stuff. You have drugs that can alter protein synthesis. You can have drugs that will block this hormone from starting the whole process. And then you can also have some drugs that will disrupt the signaling pathway within. And then here you go again, you've got another little thing. And I am almost at the limit of my 15 minutes, and so sorry this one was so long. Hopefully that helped. In particular on this one, to remember, don't get hung up on the words and the names. Let the pictures sink in, and I know you all let them sink in.